heterogeneous group of receptors that have a common property of perceiving the sense of touch due to contact with external solid objects, current of air and water, or due to mechanical body strains, or even in the case of internal body pressure. Some of the mechanoreceptors are specialized in responding to the displacement of air particles and behave as sound receptors, auditory organs. The principal mechanoreceptors are the tactile organs, the campaniform organs, the chordodominant organs, and the chordotonal organs, and the sensitive. Tactile organs are trichoid sensilla. They are the simple articulated sensory hairs and distributed on the entire body surface and are commonly called as the sensilla. They are made up of cuticle and articulated within the socket with the body wall. The trichoid sensillum is formed by two cells, the hair by the trichogen cell and socket by the tormogen cell. Both the cells are modified epidermal cells. Each sensillum is innervated by a bipolar sensory neuron. The dendrite of the neuron is enclosed at the base of the hair by a cuticular tubular sheath called the scopole. It is in some cases provided with a distal cap called a scopole cap. The scopole is placed at each bolt. That is scopole. The dendrite of the neuron is enclosed at the base of the hair by a cuticular tubular sheath called scopole. So these are symptoms of massive expression of the trichoid sensillum in the parts and in a type tibia and dorsal. These organs become the major tactile organs of the insect along the sense of touch. There are also some trichoid hair plates at the joints of eyes of the pages and function as proprioceptors, sliding of the segments over each other. There are tactile hair beds on the facial region of the head of the locusts and some Lepidoptera, on the neck of dragonflies and on the wing margins of Lepidoptera, which are responding to the air movements during flight, conducting 600 or more impulses per second, and thus detect efficiency and changes in air current. The tactile organs of antennae and lower segments of legs perceive earth-borne vibrations in terrestrial insects, Orthoptera, etc., and water surface vibrations in aquatic insects. The impulses are produced in the dendrite by either movement, bending, or straightening of the hair, exhibiting phasic response, or during bending alone, showing tonic response. Tactile hairs of various appendages show plastic response, while those occurring within the joints of the legs, prothorax, genitalia, are proprioceptors, and hair beds found in locusts, mantids, dragonflies, and ibis, etc. So when we try to learn about the trichoid sensillum parts, we could see here cuticle, vacuole, dendrite, epidermis, axon region, sense cell, trichogen cell, tormogen cell. Seen externally but recognized from the dome-shaped cuticular areas, oval or elliptical, elevated above or depressed below the general body surface, and are of about 20 to 30 mm in diameter. The skeletal rod or sense rod or scopole bearing the terminal filament of the sense cells is inserted into the dome-shaped area. So campaniform sensillum, axon, sense cell, accessory cell, cuticular connection, scopole, then vacuole, inner lamella, cuticular dome, outer lamella, and cuticle, vacuole. These are the regions. So the campaniform cells occur in various parts of the body and form distinct groups, often at the wing base, halters, cerci, palps, and on the base of trochanter, femora, tibia, and tarsal segments. On the halters, they form Hicks papillae, basal plate, and scapal plate at the base, While on wing base of diptera and fly empis, they form two groups along the subcostal vein and three groups along the radial vein. The mechanical stretches and the forces exerted on the cuticle by muscles and gravitational forces stimulate the campaniform sensilla. The campaniform sensilla therefore function as the proprioceptor. They respond to the mechanical stimuli in terrestrial insects, water pressure in aquatic insects, and air pressure in flying insects. The halters therefore can function as balancing organs. Then chordotonal organs. Each chordotonal organ is made up of a spindle-shaped group of scolopophores. They are intravisceral mechanoreceptors. Both the ends of these organs are attached to the integument, integumental chordotonal organs. Each scolophore consists of a cap cell provided with a terminal ligament apically and acts as a scolopale cap. The envelope cell contains sensory cuticle a liquid-filled vacuole. The entire scolopale lies in a liquid-filled medium in which moves freely. Both the cells are hypodermal cells. The sensory cells is the third cell of the scolophore, which extends dendrite process apically. It runs through the vacuole, scolopale, and finally terminates into the end knob of scolopale. So when we try to learn about the basic structure and integumental and subintegumental organs, cuticle is there, epidermis, scolopale cap, scolopale, cap cell, vacuole, envelope cell, dendrite, sense cell, and axon. Then integumental cell, subintegumental nerve. These can be seen. They occur in legs of femoral, distal tibial, and tibiotarsal regions in abdomen, various appendages, wing base, and diptera and maggots. There are, in case of diptera and maggots, there are about 90 chordotonal organs in the entire body of Drosophila larvae. They are stimulated by external pressure on the body, passive movement of the segments, tension of muscles, internal pressure changes due to blood and trachea, and are also sensitive to the mechanical vibrations. They can perceive sound and thus in many cases function as a sound receptors or may form distant auditory organs. Okay, now let's learn some demarcations of this chordotonal organ, like specialized chordotonal organs. Johnston's organ. Except Columbola and Diptera, all other insects possess the Johnston's organ in the second antennal segment or pedicel. The sensilla are radially arranged and are attached at one end to the second and third intersegmental membrane and at the other end to the wall of the second segment. The axons of the sensory cells of these organs run back and at the antennal nerve. The antennae also contain large number of campaniform and chordotonal sensilla. In Odonata, they are poorly developed, while in male chironomids and chelicets, they are enormously developed, but the other chordotonal organs are entirely absent. The Johnston's organs respond to the movement of the antennal flagellum and thus act as not merely the proprioceptor but also perceive air currents during flight. In case of excess vibrations through substratum during walking and through water then fibers, nerves, scalp region. They act as indicator and regulator of flight speed in Calliphora. They are the predominant organs of hearing in the chironomid and chelicid males, which have plumose antennae. In the aquatic bugs, Notonecta and Corixa, the Johnston's organ function as organs of orientation during swimming. In bees, the organs are called static organs as they respond to flagellar movement. The honeybee can regulate the speed of flight with the help of these organs. Okay. Then another demarcation is the subgenual organ. Subgenual organ, they are confined to the proximal part of tibial segment of the legs of the insect except Michelus, Coleoptera and Diptera. They do not occur at the joints. Each organ is made up of Each organ, that means each subgenual organ, is made up of 10 to 40 scolopedia and are provided with the attachment and accessory cells. The scolopae is connected to the attachment cell. The processes of accessory cell collectively form the attachment body. Attachment body is fixed to the cuticle at one end and the other end is supported by a trachea. The subgenual organs are highly developed in Odonata, Dictoptera, Orthoptera. In these groups of insect, proximal and other distal subgenual organs are present. The proximal subgenual organ is the true subgenual organ, while it at least Tetygonidae and Grilloidea. The distal subgenual organ is modified into the intermediate organ and the crista acoustica. In Hemiptera, Neuroptera, and Lepidoptera, the subgenual organs are poorly developed and are in fact the distal subgenual organs. Sensory nerve is there, trachea, sensus cells, hemocoel, scolopae, body wall, accessory cell, and attachment body. The subgenual organs are sensitive to the vibrations of the substratum and airborne sounds of high intensity. In Periplaneta, they respond to the vibrations at frequencies up to 8 kc per second with an optimum of 1500 c per second. The next demarcation is auditory or tympanal organs. First, Johnston's organ, subgenual organ, then auditory or tympanal organ. These are paired modified chordotonal organs of hearing. Each organ consists of a thin cuticular membrane and a tympanic membrane or tympanum backed by tracheal air sac and a group of chordotonal sensilla attached or closely lying adjacent to the inner surface of the tympanum. The chordotonal organ is commonly composed of scolopodia ranging from 1 in Plea Heteroptera to about 1500 in Cicadidae. They are of two types. The chordotonal organ is commonly composed of scolopodia ranging from 1 in Plea to about 1500 in Cicadidae. They are of two types, integumental found in Acridiidae, Cicadidae and Lepidoptera, and subintegumental found in Grillidae and Tetygonidae. The tympanal organs are developed independently and thus they differ in their location among different groups of insects. They are found in prothoracic legs, mesothorax, metathorax, abdominal second segment, wing base. The tympanal organs of Coryxa, Plea, Nepa, etc. are rudimentary. So let's try to learn about the structure of this tympanum. Longitudinal section of tibia, a foreleg of Dactychus showing tympanum. We could see sugenual nerve, tympanal nerve, supratympanoid organ, intermediate organ, crista acoustica or tympanal organ, scolopedia, supporting band, inner the displacement receptors responding to the disturbances of much lower intensity. They function by perceiving the pressure pulses which develop from the sound source by perceiving displacement of air causing vibrations of the earth membrane. The sound nerve waves cause it to vibrate. The vibration moves into the chordotonal sensilla and the impulses are produced in the auditory nerve. The tympanum collects the energy of sound as it is the most dedicated membrane and cannot be displaced easily since air-filled cavities behind it reduce the damping. The air cushions improve mobility of tympanal membrane and amplify the bands and thus improve the auditory sensitivity. The sounds are discriminated by the rhythmic variation in intensity which make up the modulation pattern and not by variation in pitch. The insects are, however, unable to discriminate between the sounds of different frequencies. The sound-bearing insects are most sensitive to the high-frequency stimulation. The auditory range of tympanal organs is as follows. Acridids 300, locusts 600 to 45,000, cicadas 100 to 15,000. While in man, the original frequency is 1,500 C per second. The characteristic of sound hearing of the insects is that they are able to locate source sounds. The time constant of the tympanic organs of insects is about 2 milliseconds or even less in comparison to that of human ear, about 20. Due to such time constant, man cannot distinguish the sounds following one another at the intervals of less than about 1 by 10th of a second, where insects are able to distinguish sounds separated by intervals of even 1 by 100th of a second. The insects proprioceptive organs, maintaining the equilibrium of the body and in orienting the insect with respect to gravity. Basal plate, basal lobe, scapels and stack region, end knob. These Hicks papillae. These are the parts. The larvae of aquatic insects, Limnophilic Phascipennis, bears a pair of sacs in the last abdominal segment. Its sac is provided with the sensory hair at its blind end and granules or statoconia in the lumen. The sac is filled and emptied with water rhythmically by pumping action, which causes the statoconia to fall upon the sensory hair. On the other insect, Catocoptera fascicula statocyste, containing movable statolith in the abdominal 10th and 11th segment. The nymphs of Nepa contain specialized static organs in the form of hairy sensory apparatus along the ventral surface of the abdomen, containing a film of air. In adults, the apparatus is confined closely to the spiracles of 3 abdominal segments. They function as differentiated manometers and these adult bugs contain a pair of oval depressions on the sternum of second abdominal insect or segment close to spiracle. Next is about chemoreceptors. So we completed mechanoreceptors. There are different types like common chemoreceptors, gustatory chemoreceptors, and olfactory chemoreceptors. And on the basis of morphology, the chemoreceptors are often grouped into 4 types. Next 4 types: Sensilla trichoidea, olfactoria, Sensilla basiconica, then Sensilla coeloconica, and Sensilla placoidea. Okay, placoidea. So different parts are there: thin-walled seta, cap cell, cuticle, thermal dendrite strand, then enveloped cell, fascicle of dendrite, sensory cell, dendrite, sensory cells, nerve, scolopale, cap cell, epidermis, different parts. So Sensilla trichoidea, olfactoria. These are thin-walled hairs which have been described from the tarsi and labella of tabanus, tarsi of musca, and antennae of several species. They are the sensory cells provided with hairs. Then Sensilla basiconica and styloconica. Sensilla trichoidea, olfactoria. Next, Sensilla basiconica and styloconica. They are peg-like or cone-like organs occurring on antennae of many species, palps of Hygrophilus paces, Agroitus larvae, antennae and palps of Periplaneta, Lepidoptera larvae, antennae of mysids, palps of butterflies, hypopharynx, epipharynx, and on surface of labrum of many insects. They are the sensory cells provided with pegs. In Sensilla coeloconica, the cuticular process is sunk between the general level of integument. The organ is very deeply sunk and connected to the surface by an elongated tube, Sensilla ambulacea. They found on the antennae of various insects, Coleoptera, Lepidoptera, and Hymenoptera. They are the sensory cells provided with pits. Pit opening, cavity, cuticle, vacuole, fascicle, envelope cell, sensory cell, nerve. Sensilla coeloconica. And Sensilla placoidea. Each consists of a thin cuticular plate, approximately circular or elongated oval, beneath which lies a capsule and envelope cell. The latter is penetrated by bundles of distal process from the bipolar neurons. The process coils in a terminal strand which ends in the pore plate. Pores, capsule, envelope cell, epidermis, sensory cell, fascia, vacuole, terminal dendrite, outer plate. Antennae of many insects. Aphidoidea, Coleoptera apis, bear these structures. They are the sensory cells provided with plates. Physiology of keen insect. Keen insects, they are not subject to distinction between such as taste and smell. Both types of olfactory sensory sense of vehicles Gustatory refers in the terrestrial and aquatic insects, and former is a sense of solutes, while in the later it is perceived through the liquid substances. The gustatory, so taste and smell are not different in case of an insect. Okay, and the gustatory and olfactory chemoreceptors often differ in their location in the terrestrial insects. In the aquatic insects, moreover, the Sensilla basiconica, situated on the tip of antennae, are the principal chemoreceptors for both gaseous and liquid stimuli. Okay, then gustatory receptor. In the terrestrial insects, the gustatory chemoreceptors are mostly confined to the mouthparts, wall of true oral and hypopharyngeal cavity, rarely on the tarsi and distal end of the tibia, as found in the butterflies' mouths. Dipteran flies, Calliphora musca, and the honeybee, Apis mellifera, and in a few insects such as the ants, bees, and wasps on the antennae. Deisticus, the aquatic beetle, is capable of distinguishing the taste of four quantities such as sweet, sour, salt, and bitter, while ants, bees, and wasps can differentiate plain and sweet alcohol, plain and sweet. Sensory hairs, which are specialized independently for perceiving the sense of the cations, anions, sugars, and water. Presence of large number of salt receptors seem to be a common feature among several insects. These are sugar receptors specialized in the qualitative as well as quantitative differentiation of the sugars. The labillar hairs and the pharyngeal receptors in Calliphora can taste about 15 sugars by the basiconic sensilla. Similarly, in some insects, the tarsal and antennal organs of taste are very sensitive to sugars. Then taste sensitivities. Detier and Chadwick in 1950 reported acceptance of sugars on the following sequence: B mouth parts, Calliphora mouth parts, Formia legs. And all these insects, sucrose, maltose, and trihalose, and alpha glucose, and used to stimulating Food selection in insects is carried out almost exclusively by chemoorientation, mostly by the test. The polyphagous insects feed on only a few kinds of wide variety of substances such as acids, salts, alcohol, ester, amino acids, and oils. All are rejected at high concentration, but dilute concentration of acid and salts are sometimes preferred. Then electrophysiology of the gustatory receptors. Most of the electrophysiological studies are made on those of the Formia's labillar gustatory receptors. The findings are given below. The spike potential is always initiated near the Then the action potentials from cation receptors and sugar receptors appear to be the efferent activity at the greatest level during few initial seconds of stimulation. The latent periods elapsing between the instant chemicals applied to cation and sugar receptors are short, usually of a few milliseconds. The 45 minutes per second m per second sustained potential is in fact generator potential. The adaptive curve shows that activity is often highest during the initial few seconds of stimulation. The most significant part of the information is reaching the brain within a short interval. Similarly, the relative curve shows the usefulness of receptors in differentiating the quantities as well as qualities of the stimulating chemicals. Then olfactory reception. They include mostly the placoid, basicoid, coeloconic and trichoid sensilla situated on the antennae of large number of insects, palps of the butterflies and the label of the blowflies. The olfactory threshold is very low for some substances like skatole at a concentration of 0.003 mole per liter of air can attract geotrophs. The sense of smell is stimulated by a low concentration of vaporous substances which are volatile at the ordinary temperature. If this mellifera has some thresholds to that in man for various orders of essential oils. Various factors affect the olfactory threshold: age, sex, nutritional status, habitat, temperature, humidity, and the rate of airflow. Age-dependent effects can be seen in Pimpla ruficollis, a parasite of pine shoot moth, then Eutria boeliana, showing repulsion to pine oils when immature, then attraction when mature, so that they can oviposit in their hosts. The next classification of odors. Odors may be classified as sexual attractants, recognition odors, oviposition attractants, and food attractants. Okay. Sexual attractants. Male gypsy moth, Lymandria disper, is attracted by the odors ketol and visparlure from the over 2 miles by a virgin female of its own species. Recognition orders. Social insects can detect members of their own species, castor colony, by olfaction, termite, and honeybees. The oviposition attractants. Parasitic insects are attracted by sense of smell from their hosts towards the site of oviposition. Insects with sarcophagus larvae are attracted by odors such as ammonia, skatole. Female Habrobracken discovers its host aphasia by smell through antennae. Then food attractants. Phytophagous insects are attracted to their host plants by the smell of essential oils. The fruit-sucking moth, Othris materna, from the ripe orange fruits. Mechanism of chemoreceptor olfaction. Various hypotheses have been put forth in order to explain the mechanism of olfaction. One hypothesis of smell is that many odor substances absorb in the infrared and show a Raman shift. Light waves are scattered by these substances and the cells are stimulated. Absorption of heat from epithelial cells by the odorous agents cools the epithelium. Another hypothesis is that the chemical stimulation causes liberation of a nerve excitement, acetylcholine, which stimulates the olfactory receptors by depolarizing their cell membrane. According to a recent hypothesis, the olfactory threshold is related to a critical number of odorous molecules absorbed within a given area of the cell membrane and to the adsorption constant for passing from air to the oil-water interface. Recently, Vogt described the molecular mechanism of olfaction. The mechanism of olfaction is carried out by two processes. First is receptor process, transport of odor molecule, and the second is the transduction process, conversion of chemical to electrical energy. The perireceptor process, the odor substances are mostly lipophilic. The odor molecules are first absorbed in waxy layer of epicuticle of the olfactory receptor sensilla. They diffuse into the pore, pass through pore kettle and pore tube lying in the cuticular wall of the sensillum and release into the receptor lymph. The odor molecule molecule is then linked with the order binding protein and moves towards the receptor site of the dendrite membrane surface. The order binding proteins are investigated in the lymph of the olfactory central of both the sexes. The order binding proteins are water soluble proteins and specifically characterized in binding the order molecules only. The order molecule gets separated from the order binding protein molecule and thereafter it binds to the receptor protein molecule. The order binding protein molecule moves towards the pore tubule to carry another order molecule. The order molecule binds with only its specific receptor protein molecule and enables discrimination among a wide variety of orders. So, order particle, molecule, lipid of epicuticle, pore, pore tubule, wall of sensillum, order in degrading enzyme, order binding protein, receptor molecule, dendrite, receptor lymph. That is the perireceptor events during olfaction. Once again I'm gonna read the parts. Order molecule, lipid of epicuticle, pore ketch, then pore tubule, wall of sensillum, order degrading enzyme, order binding protein, receptor molecule, dendrite, receptor lymph. The transduction process with the binding of order molecule with receptor molecule, the second messenger system is activated immediately through the second messenger system, in which mostly the transduction process with the binding of order molecule with receptor molecule, the second messenger system is activated immediately through the second messenger system, in which mostly inositol triphosphate or sometimes CAMP is involved. A depolarization of the dendrite membrane receptor potential takes place spontaneously. Then optimal receptor potential produces an action potential of either phasic or tonic nature. Action potential is often elevated in proportion to the concentration of order above the threshold. Lastly, degradation of the order molecule is carried out by an enzyme called order degrading enzyme, and it is removed quickly from the dendrite membrane. To order degrade, which one is the order degrading enzyme? No, it's not named. Yeah, two order degrading enzymes, acetate esterase and aldehyde oxidase. Acetate esterase and aldehyde oxidase are detected in the receptor lymph in the tassar silkworm and Serratia. Then order discrimination in Lepidoptera, Spodoptera and Helicobacter. The olfactory receptors are very specifically responding to the sex pheromones of host-specific orders, as they are efficiently responding to only a single order compound, and they may even show a marked selectivity for a specific isomer. Individual olfactory receptors are known to respond to different non-pheromonal order compounds, and in Spodoptera as such 21 types of olfactory receptors were determined on the antennae. In this insect's olfactory receptors is equipped specifically with only one type of order molecule, binding protein, and a receptor molecule. This is the reason that the adult butterflies and bees respond to a wide range of plant-derived compounds and collect the nectar accordingly. On the other hand, in cockroaches and bark beetles, one and some type of receptor respond to a large number of related compounds. In these insects, olfactory receptor contains large number of olfactory molecules, binding proteins, and a variety of receptor molecules. In gestation, the contact chemoreceptors function as the gestatory receptors and situated densely on the mouthparts, labrum, maxillae, labium, epipharynx, hypopharynx, oral cavity, legs, tarsi, and sometimes in the tip of antennae, and in some insects even in all over surface of the body. During feeding, the mouthpart gustatory receptors first get immersed in saliva or food fluids and then respond to the present in the solution. The contact chemoreceptors present on tarsi normally respond to the chemical spread on a dry surface of leaf. The compound sugar or salt binding proteins present in a fluid concentration at the tip of sensillum serves as carriers for the compound spread on the leaf surface. and the substances on tip of sensilla of the labial palps in Periplaneta. The proboscis and tarsi of dipteran flies contains a specific sugar binding protein. These sugar binding proteins belong to the same family of pheromone binding and other order binding proteins. Two protein molecules serving as sugar receptors have been detected in the fly Phormia. The inorganic ions, however, act directly on the ion channels in the dendrite membrane. The gestator receptors, sugar and salt receptors, may have different peri-receptor mechanism. The transduction mechanism of gestation is still undiscovered. It is generally believed that the receptor potential initially develops on the dendrite distally and then passes to the spike initial zone action potential at the base of the axon. The magnitude of action potential is directly proportional to the concentration of sugar or salt in the material came in contact. Similarly, the number of action potentials is often proportional to that of the receptor potential. The nerve cells of sensilla responding to substances spread in the habitat have been sensitive threshold than those responding to the sugars, amino acids, and other nutrients. Each neuron within a sensillum is specifically responding to a different class of compound. Mostly, some respond to salts while others to sugars. They are often called salt, sugar, amino acid, or dicterant receptors, which can even be recognized from their behavior. The sugar receptor, on the basis of their specificity, can further be classified into the sucrose, glucose, fructose, and other specific sugar receptors. Their specializations are determined by the type of receptor molecule they contain on their dendrite membrane. Let us notice that the concentrations of various substances in the hemolymph increase or decrease the sensitivity of the gestator receptor relatively. A role of some hormone released from the corpora cardiaca has also been suggested in the mechanism of gestation. It is now understood that the insects cannot taste the proteins. Now, next topic, significance of chemoreceptors. Significance of chemoreceptors. Besides serving as a sensory system for evaluating taste and smell, the chemoreceptor control various biological processes during the life of insect, among which control of feeding, oviposition, and behavior deserve special attention. Control of feeding. The chemoreceptors, which are situated on mouthparts, first make contact with the food and detect its suitability, and then the insect immersing feeding. So, continuous sensory input is essential for continuous feeding. Phytophagous and sarcophagous insects select food with the help of chemoreceptors. And control of oviposition. The female locusta initially detect the concentration or salts in sand by chemoreceptors laying on the ovipositor and starts digging a hole for oviposition only if the sand contains low concentrations of salts. Similarly, the fertilized female of Lucilia detects wool, the olfactory receptor, moving towards the site, starts ovipositing on a live sheet. And control of behavior, various intraspecific mode of communications and behavior in social insects, sexual attraction, copulatory and post-copulatory behavior in various insects, and solitary and gregarious behavior of locusts, all are chemoreceptively controlled. So olfactory chemoreceptors are from environment, they expose and receive the signals from the eye. So the most significant thermohydroreceptor, the flagellar annuli of the antennae of insects, which respond to the environmental temperature and humidity. On each antennae, the temperature and humidity sensilla are about 50 in Apis and 100 in Periplaneta. These temperature and humidity sensilla consist of a short peg and an immovable socket. The dendrites of two neurons are fused together, forming a thick dendrite cord. It is enveloped into a dendrite sheath on the distal region, and fused microtubular complex, ciliary region of the dendrite, occupies the proximal region of a peg. The dendrite cord may be concealed in a pit or cuticular cavity or exposed outside. The dendrite of the third neuron terminates below the base of the peg. The ptomagen, tricogen, and tecogen cells are well differentiated from the epithelium at the base of the sensilla. The sense cells extending their dendrites into a peg are responding to humidity, hygrosensitive, while third sense cell extending dendrite up to a base of a peg and is the thermosensitive. In some insects, Locusta and Periplaneta, the thermosensitive multiporous olfactory sensilla are notified on the antennae and about 100 such sensilla on each maxillary palp also. Change in ambient humidity may stimulate the dendrites of the peg directly and often have a higher tonic firing rate. The thermoreceptors are responding to even a less than 1 degree Celsius change in temperature and have higher firing rate in response to fall in the temperature and are called cold receptors. Cave dwelling beetle Spheophysis. In that insect, cold receptors are seen. In cockroaches, however, the olfactory thermoreceptors respond to change in both the higher and lower temperature. Then thermoregulatory mechanism. In majority of insects, all optimal physiological processes including enzyme activity occur at a relatively high temperature at the range of 30 to 40 degrees Celsius. The body temperature of an insect is in fact an indication of ambient conditions coupled with the metabolic heat produced, which is often elevated significantly by the activity of thoracic muscles during flight, endothermic heat gain within the body and from the solar radiation, exothermic heat gain from the environment. Similarly, evaporation is the main source of heat loss in the terrestrial insect, which releases latent heat of evaporation outside and cools the insect body. Heat loss is directly controlled by the environmental humidity. At high humidity, little heat is lost, while at low humidity, more heat is lost through the evaporation process. The ambient temperature of insect body is often balanced between the quantum of heat gain and heat loss. So cuticle is there, peg is there, dendrite of hygrosensitive neurons, then dendrites of thermosensitive neurons, dendrite sheath, thecogen cell, tricogen cell, tormogen cell, neuron, somata, then axon. These are the parts of thermohygroreceptor. Cuticle, peg, dendrites of hygrosensitive neurons, dendrites of thermosensitive neuron, dendrite sheath, thecogen cell, tricogen cell, tormogen cell. Okay. So the insects are generally not capable to maintain a constant body temperature, but still some species can regulate their body temperature at optimal level by undergoing some behavioral and physiological adaptations. The nymphs of locust Schistocerca have become very active at 44 degrees Celsius, approaching upper lethal temperature, and move from unfavorable to the favorable low temperature area and save their life. In these insects, most favorable optimal range of environmental temperature is 35 to 45 degrees Celsius. Some flies, grasshoppers, caterpillars, and butterflies move into sunlight during low environmental temperature or move into dark when the air temperature is high and able to perform their activities at maximum level. One of the best example of physiological adaptation for thermoregulation is presented by the dragonflies. The dragonfly annex reduces wing beat frequency and flight speed and adapts to gliding when air temperature rises from 20 to 30 degrees Celsius. In honeybees also, similar mechanism for regulation of thoracic temperature is noticed. Most of the flying insects control their body temperature during flight by regulating heat loss. Abdomen acts as a radiator of heat exchanger in the moths and bumblebees. As the excess heat is produced in the thorax, heart beats rapidly and increases the rate of hemolymph circulation to abdomen and reduces the efficiency of the heat exchanger. Some temperate insects such as the moth, Upsila fly even at an ambient temperature of 0 degrees Celsius by raising their thoracic temperature by 30 degrees Celsius. The thorax of the moth is insulated by a hair pile and a well-developed countercurrent heat exchange system. Several insects can survive at extreme low temperature as their supercoiling temperature ranges from 5 to 10 degrees Celsius. Such freeze-tolerant insects, cockroach, Cryptocercus, butterflies, papilio, beetles, etc. Once frozen, then able to withstand low temperature for a low period, and particularly some other caterpillars can survive even at minus 70 degrees Celsius for 9 months. Survival in frozen insects is possible because of presence of some supercoiling agents such as the ice nucleating proteins and lipoproteins in the hemolymph or crystals of calcium phosphate or uric acid in the Malpighian tubule, which prevent intracellular ice formation. In addition, glycerol is the principal cryoprotectant cryoprotectant in insects, which controls osmotic pressure of hemolymph and helps in regulation of the cell volume during extracellular ice formation and stabilization of protein. The supercoiling protein point varies in the Arctic insects, minus 35 degrees Celsius in the eggs and minus 26 degrees Celsius in the larvae of British apron. Most of these Arctic insects are susceptible to supercooling, not supercooling agents. Okay, supercooling. I'm sorry, supercooling as it is varying inversely with the accumulation of polyhydric alcohol, and insects die whenever exposed to high temperature than the supercoiling point. Main reason for death at high temperature seems to be the protein degradation, metabolic imbalance, and accumulation of toxic substances. Then specific properties of thermosensitive sensilla. The thermosensitive neurons of antennal sensilla in insects respond to both convective heat and thermal radiation. Thermal radiation is an exclusive orientation due to some insect species. In some cases, thermosensitive sensilla detect thermal radiation very meticulously. The pit organ of Australian fire beetles carries up to 80 thermosensitive sensilla. Accessory structure of these sensilla change on adaptation of thermal radiation emitted at wavelengths of 35 in forest fires, resulting in a photomechanical stimulus transduction. In the physiological temperature range of 30 to 35 degrees Celsius, corresponding to longer wavelength of about 10 am. However, thermal radiation has much less power, as indicated in the Stefan-Boltzmann law. Therefore, the detection of difference in thermal radiation in other particular temperature range poses high demands on the sensory system. Blood-sucking bugs, Priatoma infestans, and Rhodnius prolixus are guided solely by the thermal radiation emitted from their endothermic hosts. That means whatever the host emits, they catch on that. These bugs seem to measure the distance to their host based on temperature gradients along their antennae, and probably evaluate the size of the object by saccadic antennar movements, like the triatomine bugs. Leaf cutting ants can use thermal radiation as a cue for orientation. Similar to their detection system in the bugs, ants scan their environment by fast antennal movements to detect heated objects and use them as learned landmarks. During antennal scanning, the antennae of leaf cutting ants can reach angular velocities of up to 3.6 degrees Celsius ms inverse that may generate rapid minor temperature transients at thermosensitive sensilla. Because the sensilla Coleoconica of leaf-cutting ants, Atta Woolenweideri house a thermosensitive neuron. They are good candidates for providing the ants with the underlying thermal information used for orientation. Irrespective of whether thermal radiation and or convective heat act as an antennal sensilla, the resulting temperature transients are expected to be minute and brief. The thermosensitive neuron very quickly respond to a drop in temperature with a brief phasic increase in spike rate and thus classifies as cold receptor. The short latency and brief phasic response enable the thermosensitive neuron to follow temperature transients up to an estimated frequency of around 5 Hz. Although the neuron responds as a cold receptor, it is extremely sensitive to warm stimuli. A temperature increase of only 0.005 degrees Celsius already leads to a pronounced decrease in the resting activity of the thermosensitive neuron. Through sensory adaptation, sensitivity to temperature maintained over a wide range of ambient temperature. The thermosensitive neuron of Sensilla coleoconica is adapted to detect minute temperature transients, providing the ants with thermal information of their microenvironment, which they may use for orientation. So, in this chapter, we mentioned the different receptors or the sense organs like mechanoreceptors, the tactile organs, or trichoid sensilla, then the campaniform sensilla or organs, chordotonal organs, specialized chordotonal organs like Johnston's organ, then subgenual organs, then auditory or tympanal organs. Later we mentioned static organs. Next, the chemoreceptors types: common chemoreceptors, gustatory chemoreceptors, olfactory chemoreceptors, sensilla, trichoidea, olfactoria, Sensilla basiconica, styloconica, then Sensilla coeloconica, and Sensilla placoidea. Gustatory reception, taste activities, then electrophysiology of gustatory receptors, olfactory receptor, classification of orders, sexual attractants, recognition orders, oviposition attractants, food attractants, mechanism of chemoreception, olfaction, the perireceptor process, order discrimination, gustation, the transduction process, significance of chemoreceptor, control of feeding, control of oviposition, control of behavior, then thermohygroreceptors, structure, thermoregulatory mechanism, then the specific properties of thermosensitive sensilla. So we mentioned later we mentioned thermohygroreceptors, the specific properties of thermosensitive sensilla. Thank you for learning with me.